Hello, welcome to my channel, AI Medical School. This video topic is acute inflammation. So what is inflammation? Inflammation is the reaction of living tissues to all forms of injury. It is the reaction of vascular and supporting elements of a tissue to injury, and results in the formation of a protein-rich exudate, provided the injury has not been severe enough to destroy the area. Advantages of inflammation. Inflammation is essentially a protective response intended to destroy invading microorganisms, inactivate toxins, and achieve healing and repair. Disadvantages of inflammation. Although inflammation helps clear infection and along with repair makes wound healing possible, both inflammation and repair may be potentially harmful, such as swelling of the brain, caused by acute inflammatory response against a viral infection, may lead to death. Types of inflammation: acute inflammation and chronic inflammation. Acute inflammation. Acute inflammation is the immediate and early response to injury. A critical function of this response is to deliver leukocytes to the site of injury, where they help clear invading infectious agents, as well as degrade necrotic tissue. Acute inflammation is regarded as the first line of defense against injury. Causes of acute inflammation: mechanical trauma, chemical injury, radiation therapy, such as heat, ultraviolet light and ionizing radiations, injury to cold and heat, injury due to microorganisms, for example bacteria, viruses, fungi, and parasites, injury due to an immunological mechanism. Characteristics of acute inflammation. Acute inflammation is characterized by relatively short duration, lasting from a few minutes up to a few days. Formation of inflammatory exudate, predominantly neutrophil, leukocyte, and accumulation. Changes in acute inflammation, vascular changes, and leukocytes. Cellular events. Vascular changes are changes in the caliber of the blood vessels, changes in blood flow, increased vascular permeability and fluid exudation. Leukocytes. Cellular events are margination, adhesion, emigration, chemotaxis and leukocyte activation, and phagocytosis. Let's discuss it one by one. First, vascular changes. Changes in caliber of blood vessels. Initial vasoconstriction. The earliest response to an acute injury is the transient constriction in the small blood vessels. Persistent vasodilation. Vasodilation rapidly follows the initial constriction and persists for the duration of the inflammatory process. Change in blood flow. The first change is an increase in blood velocity due to dilatation of the terminal arterioles. This phase is for a short time and is followed by the slowing of the bloodstream. The main factor in the production of this stasis is an increase in vascular permeability. Fluid is lost from the blood, and the blood viscosity is increased that slows down blood flow. Increased vascular permeability and FULID EXDATION. The permeability of capillaries and venules is a function of the intercellular junctions between vascular endothelial cells. These pores normally permit the passage of small molecules. In acute inflammation, there is an immediate marked increase in the permeability of capillaries and venules due to the active contraction of actin filaments in endothelial cells. As a result, widening of intercellular junctions occurs, which allows passage of a large amount of fluid and high molecular weight proteins. This process is called exudation. This exudation of fluid produces tissue swelling, called inflammatory edema. Mechanism of increased vascular permeability. There are 5 possible mechanisms of increased vascular endothelial permeability. 1. Endothelial contraction, due to actin filament contraction in endothelial cells. 2. Endothelial cell retraction. 3. Direct endothelial injury resulting in endothelial cell necrosis, increasing the vascular permeability, such as in burn and infection. 4. Leukocyte-mediated endothelial injury. Endothelial cells may occur from the proteolytic enzymes released from the leukocytes which accumulate during the inflammatory response. And 5. Leakage from regenerating capillaries during wound healing because new capillaries are formed which are leaky initially. Mechanism of fluid exudation. In normal tissues, the walls of the capillaries and venules are freely permeable to water and electrolytes, but not to proteins and other large molecules. The rapid exchange takes place between intravascular and extravascular water, and about 70% of the water in the blood crosses the vessel wall every minute and is replaced by water from the interstitial space. There are 3 factors that regulate this transport. Capillary hydrostatic pressure. It tends to let the fluid out of the vessel. The osmotic pressure of plasma proteins. It tends to hold the fluid inside the vessel. The osmotic pressure of proteins in interstitial space. It tends to pull the fluid from vessels to interstitial space. During inflammation, fluid exudation occurs due to increased capillary hydrostatic pressure due to enhanced blood flow, increased vascular permeability allowing plasma proteins to enter interstitial space where they exert osmotic pressure and draw more fluid from blood vessels. Exudate. It is the inflammatory extravascular fluid that has high protein concentration, much cellular debris which leaks from the vessel into the interstitial space due to increased vascular permeability, such as pleural effusion due to pneumonia or tuberculosis. Transudate. It is an ultrafiltrate of blood plasma with low protein content resulting from increased vascular hydrostatic pressure. Here, the permeability of the endothelium is normal, such as in renal failure and cardiac failure. Leukocytes. Cellular events. The critical function of inflammation is the delivery of leukocytes to the site of injury to combat infection. Neutrophils or lymphocytes, macrophages and plasma cells are dominant in chronic inflammation. Now the types of inflammatory cells. Neutrophils. The neutrophils are the most prominent cells in acute inflammation, especially during the first 24 hours. Monocytes and macrophages. After 1 to 2 days, neutrophils are replaced by monocytes and macrophages. Monocytes and macrophages are also involved in chronic inflammation. Lymphocytes. These are prominent in viral infection, and along with macrophages and plasma cells, they are involved in chronic inflammation. The last type of inflammatory cell is eosinophils. These are prominent in allergic reactions and parasitic infestation sequence of cellular events. Margination, adhesion, emigration, chemotaxis, phagocytosis. Now take the overview of what we had learned in this video. Summary of acute inflammation, changes in acute inflammation, vascular changes, initially vasoconstriction followed by vasodilation, increased blood flow, increased vascular permeability causing exudate formation and inflammatory edema. Leukocyte cellular events. Margination of leukocytes to the periphery of the bloodstream. Adhesion of leukocytes to the endothelial cells of the vessel. Emigration of leukocytes from vessel to the interstitial space. Chemotaxis of leukocytes to the affected tissue by some chemical mediators such as antibody immunoglobulin G and complement C3b. Phagocytosis of invading organism by neutrophils and macrophages, then killing and degradation of these organisms through oxygen-dependent and oxygen-independent mechanisms. Role of chemical mediators. There are a number of chemical substances produced by plasma or produced locally by the cells. They are involved in vascular and cellular events of inflammation. Systemic effects of inflammation. These are the effects of inflammation on the body detected clinically, such as fever, anorexia, weight loss, or by laboratory investigations, for example leukocytosis and raised TSR. Last, the fate of acute inflammation. Acute inflammation may completely resolve, may become complicated such as scarring or may lead to chronic inflammation. That's it for now. I hope you like it. Thanks for watching, and don't forget to press subscribe button.